Today's objectives are to identify how DNA can be cut, separated, and copied using a series of DNA manipulation techniques. Scientists use different techniques to extract DNA from cells, make unlimited copies of DNA, cut DNA into smaller pieces, and identify the sequences of bases in a DNA molecule. Alright, the first way that we're going to manipulate DNA is by cutting very large DNA sequences into smaller fragments. A lot of times DNA is way too large to be analyzed, so we're going to use these restriction enzymes to cut DNA sequences at specific sequences within the nucleotides. So if we take a look here, here's our restriction enzyme behind the DNA molecule where the sequences are, and it's going to look for a specific pattern of sequences to make cuts. So this is just zoomed in a little bit more. Here's our DNA sequence. Here are the sequences that the restriction enzyme is going to recognize. And what it's going to do is, after it recognizes those sequences, it's going to cut it into smaller, manageable pieces. That's going to allow us to separate it later on. So how do we separate the DNA? We use a process called gel electrophoresis. We'll place some DNA fragments in these little small wells at the beginning. We'll add some power to it, and we will watch the DNA travel. The larger pieces will stay towards the well. The shorter pieces will travel further away from the wells. Here is a picture of a gel that we applied some black light to, and we have our wells, and you could see several pieces of DNA is traveling down those gels, and the smaller ones are going to get further away. This is just a second set of wells where we could do more of the same type of testing. But as you can see, there are certain wells where the smaller pieces travel further than other wells. Like these are very large pieces of DNA because they're not traveling so far away from the wells. Where down here, where you can see little faint pink streaks or smears, these are very small pieces of DNA that traveled further away from the gels. Our last way of manipulating DNA is by making copies of the segments or fragments of DNA that we want. And we do this by a process called polymerase chain reaction, or PCR. It's a technique that allows biologists to make copies of genes. So if we have a certain gene that we want to make copies of, we can make copies of it based on this PCR process. So small amounts of DNA can be multiplied, making it easier to analyze. So in PCR, polymerase chain reaction, we have a wanted gene. And here's our template DNA for that wanted gene, and it's going to split apart. And when it splits apart, we're going to have nucleotides in this solution that will help fill in the complement to that strand. So we go from one to it splitting and having 2 copies. And then we're going to eventually get 4 copies in the next one, and 16, and it's going to grow exponentially. So this is kind of how PCR works. And why would we want to do PCR? We have this wanted gene that we want, and maybe we want to do some gene therapy to some cells that we pulled. In order to make this worth our while, we'll want copies of this gene that we want to work with, and maybe we can introduce it to a bacteria cell or something like that, and we can learn how to grow serums or medications and like insulin. So this would be a reason why we would use polymerase chain reaction. So there are 3 ways that we went over in today's objectives that DNA can be manipulated. It can be cut by restriction enzymes that are going to take a long piece of DNA and make it into shorter pieces of DNA. It could be separated during gel electrophoresis, and the reason why we would want to do that is we have a certain gene that we're after, and after we cut up the DNA, we could find that gene by separating it during gel electrophoresis. We can grab that gene and then make tons of copies of that gene using a technique called PCR, polymerase chain reaction, which then we could use many of those copies to help us investigate ways to do gene therapy or make medications and things like that. If you have any questions on the 3 ways to manipulate DNA, please bring them to class.